The animal kingdom is just staggering. We're talking millions of species, from the simplest little sponges to, well, us. But how in the world did all this incredible diversity happen? And how do we even begin to sort it all out? Well, today we're diving in. We're gonna go on an evolutionary journey to figure out the very blueprint of animal life. Right? It seems like an impossible task, making sense of millions of different animals. But here's the secret. Biologists are kind of like evolutionary detectives. They don't get distracted by fur or feathers right away. Nope. Instead, they look for these deep, fundamental clues in an animal's basic body plan. That's what reveals its true place in the grand family tree. So, our journey today is going to follow that exact roadmap. We'll kick things off with the basic blueprint, the rules that apply to all animals. Then, we'll trace the major evolutionary leaps, the move from simple cells to complex tissues, the absolute game-changing invention of a body cavity, and finally, the story of the chordates. Yep, that's the group we belong to. Okay, first things first. Before we can start sorting animals into their family groups, we need to get a handle on the fundamental characteristics that biologists look at. Think of these as the rules of the game that define every animal's basic design. And one of the very first clues is symmetry. It's a really simple idea, but it's incredibly powerful. You've got animals like sponges that are asymmetrical. There's no real pattern to them at all. Then you have others, like a jellyfish, with radial symmetry. That means they're organized around a central point, kind of like the spokes on a wheel. And finally, there's bilateral symmetry, a clear left side and a right side. That's the plan for most active animals, including you and me. Now, this table right here, this is basically our core toolkit for classification. Just look at those three key rows. First, we check the level of organization. Are we talking about a simple collection of cells, or are we dealing with full-blown organ systems? Next, we look at the embryonic germ layers. Is the animal built from 2 layers, making it diploblastic, or 3, which makes it triploblastic? And last but not least, we look for a special body cavity called a column. This checklist is going to be our guide for the rest of this journey, so keep it in mind. All right, let's start our journey at the very, very beginning, the dawn of animal life. This is where we find the simplest critters, which are basically just collections of specialized cells all working together. The really key thing to understand here is that these animals don't have true tissues. Yeah, their cells have different jobs, some for feeding, some for structure, but they aren't organized into coordinated layers like our skin or our muscles. This was life's very first and actually very successful experiment in being multicellular. And the perfect example of this is the phylum Porifera, which you probably know as sponges. If we pull out our blueprint, we can see they check the boxes for a cellular level of organization, and they're mostly asymmetrical. They are just beautiful in their simplicity, and they represent the absolute foundational branch of the animal family tree. Okay, now we get to the first major upgrade in animal evolution. And let me tell you, this next step was a total game changer. It allowed for things like coordinated movement, sensation, a whole new level of complexity. This was huge. I mean, for the first time ever, cells are organized into true tissues that come from 2 embryonic layers, an outer ectoderm, think skin and nerves, and an inner endoderm, which forms the gut. This new design made it possible to have a nervous system and a digestive cavity, which completely changed what an animal could do. So who were the pioneers of this new design? That would be the Phyla which you might know as Cnidaria, we're talking jellyfish and corals, and Ctenophora, the comb jellies. Both of these groups show off this new tissue-level organization, and you'll notice they typically have that radial symmetry, which is just perfect for sensing what's going on around you from all directions. But of course, the blueprint just kept evolving. The next massive step was adding a third germ layer, the mesoderm, which got sandwiched right between the outer ectoderm and the inner endoderm. I mean, think about what a huge deal this was. This new middle layer was the raw material, the building block for developing true organs, things like muscles, hearts, and kidneys. And our first triploblastic animals are from the phylum Platyhelminthes, the flatworms. They also mark the big debut of bilateral symmetry, which goes hand in hand with active directed movement. But there's a catch. Even though they have 3 layers, the space between their organs is just packed with tissue. They don't have a body cavity, which makes them acoelomates. And that's important for our next chapter. This brings us to what I love to call the column revolution. Because believe me, the evolution of a fluid-filled body cavity wasn't just some minor tweak. It fundamentally revolutionized animal design and just blew the doors open for incredible new diversity. So, what is a colome exactly? Well, it's an internal, fluid-filled space, and it's completely lined by that middle mesoderm layer. This wasn't just empty space, it was a total breakthrough. It gave organs room to grow and move, it acted as a shock absorber, and it could even work as a hydrostatic skeleton to help the animal move around. It's a truly brilliant piece of biological engineering. Okay, so here we can see the three conditions side by side. We already met the acoelomates, right? Like flatworms, no cavity. Then in the middle are the pseudocolomates, like roundworms. They have a false cavity because it isn't fully lined by that mesoderm. And finally, on the right, you've got the eucolomates, the true colomates, where the cavity is fully enclosed. This is the winning design, the one that's dominant from earthworms all the way up to us. Dominant from earthworms all the way up to us. And once that true colon was established, man, animal evolution just exploded. This superior body plan allowed for bigger, more complex animals to thrive. It led to the unbelievable diversity we see in phyla like Annelida, the segmented worms, Arthropoda, literally the most successful animal group on the entire planet, Mollusca, Echinodermata, and Hemichordata. And that finally brings us to the last branch of our journey today, the one that leads directly to vertebrates and to us. This is the story of the phylum Chordata. So, what makes a chordate a chordate? It really comes down to three key features that pop up at some point in their life. First, a notochord, which is this flexible support rod. Second, a dorsal hollow nerve cord. That's our future brain and spinal cord. And third, pharyngeal gill slits. In fish, those become gills, but in us, they develop into parts of our ears and jaw. Now, what's absolutely essential to get is that this unique chordate blueprint provided the perfect foundation for the next great evolutionary leap, the development of a backbone. That notochord eventually evolved into the vertebral column, creating a nice protective housing for that all-important dorsal nerve cord. And that, right there, set the stage for the incredible success of all vertebrates. So there you have it. We've traced this incredible path from a simple collection of cells to an animal with tissues, organs, a body cavity, and even the internal scaffolding for a backbone. It is just an amazing story of biological innovation written over hundreds of millions of years. It really makes you wonder, doesn't it? What evolutionary leap will life take next?